Hello, and welcome to Bioconductor 2021 and this workshop session on systemPipe Workflow and Visualization Toolkit. We'll start the talk shortly, but first I need to make a few announcements. If you have a question for the speaker, please be sure to enter them into the Q&A tab under the Conversations tab on the right. If you also have the ability to upvote questions, so if you see a question in there and you also want it answered, you can click on it and avoid typing it again. If you, for some reason, want to ask a question live, please be sure to just click on the raised hands feature, and one of us will bring you onto the stage to ask your question in person. Also, I want you to notice that there are a bunch of fun emoticons at the bottom. I've seen people using these throughout the conference, and they're a great way to give feedback to the speaker, to let them know that you see the screen or to give them applause or whatever you feel like. Finally, a video of the session will be available in a couple of hours. So if you have to walk out for some reason, don't panic, you aren't going to miss anything. And now let me introduce our speakers, Daniela Cassol and Zhang. And now I will disappear.
Ready to go? Okay, great. So let's start sharing my screen. Sharing my window. Okay. So please, okay, I cannot see. Can you see my screen right now? Can you? Okay, perfect. So welcome everyone. Hello everyone. I'm so happy to see you all here today for our workshop after this incredible day in these last 3 days. So my name is Daniela Cassol, and I am a postdoc at the University of California, Riverside. And I will be presenting the introduction of this workshop and also the first part of the demonstration. After that, the second part of the demo will be presented by our grad student, Zhang. So I will share my screen is up here, so you can see here the orchestra webpage, and then up here you can search for our workshop, which is called systemPipe Workflow and Visualization Toolkit. And then if you click here on the launch, you can go already click here because it can take a few minutes. But also if you click here, that will jump to our webpage. And so up here, you can find the main vignette, and here you can find our slides. So I'll begin sharing those slides. And so as I said, our workshop is called systemPipe Workflow and Visualization Toolkit. And so first of all, if you didn't find that link, you can find here the link for the orchestra, and then all the instructions how to run, how to log in to that. But please notice one thing here: we are using our 4.1, and then the Bioconductor developed version, the 3.14. So because we are so excited about our new features, so we want to introduce the new features we have been adding in the package. So if you are using the release, may not everything will work as I'm showing here today. And then down here, you can find a few resources that work our workshop website, the Docker and the instructions how to run Docker locally if you need, and the source code for this workshop on GitHub. Okay, next. So today our presentation will include a short overview of the main framework of the package and all the core features of the packages, systemPipeR and systemPipeShiny. And I'll introduce the central concepts for building and running the workflows with our packages. After that, we'll have the hands-on session with the live demonstrations. So workflow managed systems are a great solution for some of the challenges we face in data analysis, such as increasing complexity and volume of data, dealing with multiple steps, analysis, using different softwares from different sources with specific configurations. So typically, workflow systems have built-in functionalities that facilitate and simplify the running analysis pipeline, such as parallelization, requires to be portable across computing systems, uh, um, needs to have utilities for scale the number of the samples, and also the possibility to restart and take care of the dependency and generate reports. So with that in mind, I would like to introduce our toolkit for designing and running reproducible data analysis workflows in R. So our environment consists in three major modules implemented as a Bioconductor R packages. So first of all, systemPipeR provides all the core functionalities for defining, design, and running the workflows, and also interacting with both R and command line softwares. So and as well generating publication quality analysis report. systemPipeShiny integrates extend all the features from systemPipeR and integrates a graphical user interface for managing workflows, and also allows the users to interact with the results. systemPipeWorkflows, SPW, offers a collection of pre-configured workflows templates. So this collection is organized and or GitHub organization, systemPipe, and it's open to the community for contributions. So if you have used systemPipeR in the past, we encourage you to make a pull request and share your version of the pipeline. Or if you like to contribute with a new workflow, you're welcome to do so. So systemPipeR support the most important features to design and run workflows I was just mentioned. So allowing to run the entire pipeline with a few comments. And with that, systemPipeR enables and promotes the best practice for and standards for data analysis and reproducibility. So in this figure, you can see an overview of the major features, and we'll explore those features and the more details in the next slides. So the central concept for designing, controlling, and running the workflows, it's called sysargs list. It's an S4 class list-like container. And like here in this figure, each one of these steps, step 1, step 2, step 3, it's controlled by a sysargs list instance. And then another observation here is the initial metadata is the only input necessary for the users if they are using a pre-config template we have available in our package as well. And also these initial targets are optional and not required. It's that we have that possibility is not required. And then all the downstream input and outputs files are controlled internally by this workflow object. And here in this example, we can observe we have both. We have a command line and also integrative R code. We'll explore more details soon about this. And this container is flexible and allow any number of sample or steps, so it's very easy to scale your analysis. The object also keeps track of the workflow status and the dependency graph between the steps. And we'll dive in more details about the workflow object in the live demonstration. In both environments, and here I'm referring to SPS as systemPipe as Shiny and SPR as systemPipeR, we have functionalities to generate and check the directory structure. And then in the case of SPR, it's as simple as a data folder, which you store all your raw data, a folder that contains all the parameters and config files, here called param, and a folder for all the results created within the workflow. In the case of SPS, we also have all the necessary files to generate the Shiny application as the global, the www, the config, and also the SPR directory available in this project. SPR has a simple metadata annotation system that basically consists in this example, which is a simple tabular file, or can be also in a YAML format. So in this example, we can note that the first and the second column are just the path of the initial targets file. And in this case, is a parent and a RNA-seq example. And then we have additional information as the sample name, which will be the ID for each one of those samples, and also the information about the biological replicates. And you can have any additional metadata you like here. And then also this extend to summarize experiment object. And as I mentioned earlier, the downstream input and output are controlled internally by the workflow instance. So here in this example, in this step number 1, we created some outputs, and those can be used in the next steps, establishing the connection between steps and not require any additional external input for this. So for the command line tools, we have adopted the community standard for describing the data analysis in workflows, which is common language workflow, here CWL. So CWL provides the portability, scalability across platforms and environments. And one of the biggest advantages of this integration is that it allows running systemPipeR workflows for a single instance in R, but also using the command line wrappers as CWL tools and CWL runner, and also support other languages. And can also be another advantage is can also be applied for any tools and softwares, making this very flexible and useful utility. So in the case of the CWL, it's the description is based on 2 files. The first one you can see here in green describe the command line tools with all the necessary inputs, arguments, the outputs, and the base command that is required for that particular command. Here, like we have echo and then the printing message. In the second case, the second file is the input file, which describe all the values and variables to input in that particular command line. The connectivity between CWL and systemPipeR is established by the targets file, where each one of the samples became a variable in the input file. And we also will explore more about this connectivity in the live demo, so probably became more clear how this connectivity happens. So systemPipeR has several utilities. systemPipeR has several utilities for generate scientific and technical reports, as well plot functions to visualize the workflow. So here you can see RNA-seq pipeline with some information about the workflow status, the number of the expected out files, and the dependence graph of that particular workflow. Another great utility in the workflow managed system and systemPipeR is the possibility to parallel the steps in the cluster. So it's possible to submit command line and also our process to the cluster with a computer cluster with a queue system available. So on our demo today, I will not demonstrate this feature, but I would like you to know that is available in our package. And least but not last, we have several utilities for restart the project that allows the workflow to start the project in the workflow that makes the data analysis reproducible. Also, the users can export the entire workflow analysis to an R Markdown or in a bash format, also making it easy to restart that in another project or in another environment. So with that, we can jump. That was our live presentation. Let me go here. we can jump to our live demonstration. But before we go there, I'd like to point here a few resources. So our project website or project GitHub, all the links to the packages, and then the GitHub support, the Bioconductor support page, sorry, to ask any questions or any suggestions you may have. But also, I'll take this opportunity now, because I am on the slides, to acknowledge all my coworkers in this project and all the lab members of the Girke lab, and also my PI, Thomas Girke, and all our funding resources. So now I guess we could take a break. I cannot see the presentation because I'm presenting here, but if anyone has any questions, we can take a few seconds here, or I can jump to the RStudio server. No questions? Okay, so I guess I can jump, right? So here I show before here, you can click, you can find our website. So either you can follow, if you don't want to go to the RStudio, you can follow everything I'm saying on this main vignette. I have all the code here. Actually, I have more information that I will cover here today. The idea is to this vignette to serve like a future reference. Then, as I said, you have the slides here, and then we have extra materials. Sorry. Those extra materials here, I link that in the main vignette, but you can come here if you need more information. I'm sorry.
Daniela?
Yeah.
There's a question that just popped up in the chat.
Oh, great. Can you read for me?
Yeah. Somebody, Amberish wants to know, are you using this for your routine work?
Yes. Yes. That is the idea. Yeah. Maybe more things will become clear now when I present how you can use this in your data, your routine data analysis, and how you can implement this with basically any tool or any pipeline you may like to build here. Yeah. Okay. So here. So I just click here on launch, and then I log in, and then I'm here. Can you please let me know if this is a good size, if the people can see, read okay? Can give a thumb. Okay, perfect. So the first thing we'll do here, it's so we have all the files and folders here from the workshop. So the first thing I like to do is to open, I click here on vignette. I'm talking about, you can see my mouse, right? So you open up the last one, which is called systemPipeWorkflow Workshop 2021. And that is a markdown format. And then the other material I was talking about, you can find also here the markdown from the additional materials. And then, as I said, we have quite a few initial information here, introduction, the things I was just mentioned on the slideshow, and then how to run this with Docker container if you want to try this locally. So you have all those information here. And then actually I'll jump up to the line 173 on the getting start session. So if I am here, I'll go down to the getting start session here, up here. I'll close all those tabs. And the first thing you like to do is to load the library. So systemPipeR, systemPipeShiny, and systemPipeData. And then, as I was mentioned on, and also here's some information on how to find help and our links to our GitHub and so on. And now, how I was mentioned on the slide presentation, we will check for that folder structure to have the data, the results, and so on. So for that, I prepare here. If you go back on home, you can see here we have the BioC 2021 folder. So that needs to be our home directory. So either you can click here and set this as org directory, or down here, let me double check. LS. Oh my God. Yeah, so we need to set this to BioC 2021. Okay. So once we are here, I would like us to have fun and right away to create a project and then generate the workflow and then run the workflow. So I am on line 223. And then here you see this function, which is called SPR project. So what this function does is check in this directory, check if like everything is there we need to, and then generate our workflow. And then we create an object called sol. And then now I'll check this object. You can see here is an instance of the sysargs list. This is an S4 class I was mentioned. However, there is no steps added to this workflow. Also, you can check the length of sol. It's 0, so no steps here. The other thing I would like to do is to show the hidden files here. And this function just now create this .spr project, which will store all the log information in this folder. So the second thing we are doing is importing the workflow. So the entire workflow is in this our markdown file. And then I'm using this function, I'm passing the sol, this object we just created. And then we are importing and generating the workflow from this R markdown file. So when I click here, I'm running that line. You can see a few things happening down here. So first of all, we are reading the R markdown file, and then we are importing several steps. So now we can give a close look to this object, the sol object. And then here, a few things you can already start to look at. It's the step names. So we have load workflow, which are the load library, sorry. The trimming, so I forgot to mention, this will be like a very, very simple RNA-seq workflow in a toy data. So here we have, I know this is the trimming of that data, and we'll use Trimmomatic to do that. The next step is the index, to build the index with HiSet2. And then we have the mapping, and then we are calculating some mapping statistics down here in the step number 5, and calculating the read counting for each one of those samples, and then the session info down there. So also here, again, we can check the length of the object again, the object again, and then you see we have 7 steps. The other thing quickly we can see here is the status of the workflow. So all the status of the workflow in each one of those steps. So you can see it's all pending. So that means everything is rendered here. We have access to everything. However, we haven't run this yet, so it's in a pending stage. Another quick thing we can see is, for example, for the trimming, we are expecting 16 files. We don't have any file on disk that doesn't exist. We don't have access to any of those expected files. And of course, we are missing 16. And then the other thing I'd like to highlight is mapping has 4 sub-steps. So you can see HiSet, then the same tools to sort the BEM and to make the index of BEM as well. So, okay, we examine it here, and then now we can look all the same utilities using the plot workflow function, and that will open a new tab. I hope you can see my new tab. Everything I just mentioned here, you will see in a graphical way. Plus, also you'll see the dependency graph. So, which step is depending on each step. So also here, you can see another cool thing is the main branch. For that workflow. And then all those round shapes here, I mean, this is our code base step. And then this more square shape, it's a command line step. And then everything is black. The steps name are black. So mean is pending here. Okay, let's go back, and then with one click, one command. So now it's run workflow function, and then I pass the name of the object, Cassol, and then everything, a lot of things is happening. Let me show here in the results folder. So what is happening here, it will print the step it's running, this progress bar to see how much of that it's okay, and the general status of that step. So we have success, now it's running the trimming, and then you see all the samples here. And then we ask that to have a specific subfolder for that. So it's created all the samples here in that subfolder, and now it's running the mapping. And then another thing I would like to call your attention is in the command line step, we'll have this summary. And we'll have this table with all the samples, and then all the expected, the total number of the files, how many exist. In this case, we don't miss anything, so everything will be success. So this is the status of each one of the samples. Plus here is the status, the general status of the workflow. So if you, for instance, we have a warning here, the general status will be a warning because something was wrong there. And then in the case of the mapping, we also have that status for each one of, we have a sum of the files, but then for each one of that steps, also we have the status of that. And down here was the R step. So in that case, we don't have the summary. We just have the status, the progress bar, and then the final status here. Okay, so as I promised, in three commands, we generate the project, we generate the workflow, and then we run the entire workflow. So now we can examine again that plot workflow to see how it looks like. And then we'll open again another tab. And then here you see everything is green now because everything was successful. Then you see the summary of the files. The total was green, everything was okay. Those files were created. And then the time also, how long took to run that particular step. Okay, and then another cool feature here we can do, and then if I print again, you can see here everything is green now. So we are not missing any files, and then all the steps are success. Then another utility is, I don't know if you remember, but I was talking about this hidden folder, which is the .spr project. So we collapse all the log files for each one of the steps. And then in the case of command line, each one of the samples. And then with this, so this is basically a text file, but then with this render log function, so run that, you may, let me see, I don't know if it will happen with you, but okay. Oh, no. Sometimes you need to click on the try again. So we'll open a new tab. If that happens, it's just click on try again, and then we'll open this tab where it's in our markdown format, and then I have all the steps here on the table content. And then in this case, I'll have the plot visualization from the workflow on the top of the log file. And then this is very useful when you have a very long workflow. So you can come here, you can either click here on the left, but you also can click on the image. So if I click on the image, that will jump to the log for that particular command line. So here, HiSet2. And then for each one of the samples, the structure will be always the code. So you can see the exactly code we just execute, and then any standard out or standard error we may have. In this case, for HiSet, you see this the standard out of like the percentage of the alignment for that particular sample. However, if you have any error, then it's easy to see if it was a problem with the sample, if it was a problem with the command line. So it's very useful for debugging. So I'll go back here. So as I said, with like 3 commands and 2 more additional, you can run the entire workflow very easy, and it's portable and has a lot of possibilities here. So talking about those possibilities, we can dive in more details. Does anyone have any questions? It's okay. Anyone is following? Okay. So to explore more about the design of this workflow, because here we just import that from our markdown, so everything was defined there. So let's dive more how to design that, and then here we are doing this in this interact mode. But first, you can tell me like, okay, I know you are using CWL for the description of the command lines, but I don't have time, or I don't want to learn all the details and everything from CWL. So if you say that, I would say, don't worry, because we do have functions and possibilities to very easily, and we're trying to do this in a simple way, to create those parameters files for you, that description from the command line. So here, because I just show a HiSet and mapping, I bring the class one simple command from HiSet, which you have HiSet as the base command, the output, which is the same file, the reference genome, and the input file here. So the way you need to provide that, you just provide this command line in this special bash script format. So what that means is you have here in this string, in the first line is the base command, and then each one of the inputs is separate for a new line, and then you pass the prefix and then the value of that particular input, and then just specified if the type of that input, if it's a file, if it's numeric, if it's a string. So with that, let's run this, and then I can show you. So if I type here command, you can see it's just a string, like separate by a new line, every one of the inputs. Okay, so now I have some information here to specify all the details, and then I am on the line 304. So here you can find a function called createParam. So what this function does is basically take that string, and then here I'm asking to write that description files we are creating. And then when I run this, we'll check with you a few details. I will print out the base command, so you can double check if it's that everything is right. It will print all the input files. Oh, and then I forgot to say one thing here. it's in the case of the same file, you also specify that is an output. So if you say out, then we will understand that it's supposed to be an output. And then when you look here, then you see here we are expecting that same file as an output. And then again, we are printing the exactly command line, so then you can double check again if everything is okay. If everything is correct, you press 1. If anything doesn't sound okay, you can press 2 and then reorganize your string. So I'll press 1 here. And then all those CWL files is written in this specific folder. So now you can have access. So now we just create everything you need from system for CWL, and then we can start to create our workflow. And to do that, I'll jump a little bit down here on the project initialization. So I'll discuss a little more details about the SPR project. So with SPR project, as I mentioned before, this function, you can pass the location on where you want to create your project. We recommend to be your current project, especially if you want to run that, but you can create a project for any location. And then once you do that, also the other thing is to create dialog files. So here I have this SPR project, but you can name these as you like. And then the other file here I'm specifying is this YAML file, this YAML format, which is basically I'm saving the entire object and this flat file database. So that would be super, and that is in a YAML format. So that would be super simple to recover and then to restart the workflow later on if anything happens. So all the information is safe here. And every time we update this SAO object, that information will be saved also in this flat file database. And then I mentioned earlier you want to have the data for the I haven't shown, but then here if you click on data, you have all the FASTQ files. More than actually we just select 4 to run, and then you have the reference genome as well. And then here on param we have all the folder from CWL if all the tools we have available here, and you can create and customize the tools you like. And then in the results folder, then we'll jump all the results, the AL files we create on the workflow. So that also helps to keep organized and the data, the raw data separate from the AL files from your workflow. So SPR project also allows you to give another name. If you don't like those names, you can name those folder in a different way. And I guess here is my favorite thing about SPR project, which is when I'm creating this, when I'm doing the initialization of this project, I can choose either the global environment, or I can create a new environment. And the very nice thing about that, you can see my global environment here. I just have those 3 objects I just create, but I create some R objects when I run the workflow. So that is in a new environment, and keep your current session clean, and then that avoid to overwrite anything you may have in your current environment. And I guess that is a very powerful thing to keep everything separate. However, you can have access to those files, those objects, right? So we have a function called view envir and then sol. and then I pass the name of my object, my workflow, and then you can see the object, and then all those objects we just create in the when we run the workflow. And then if I want to copy those files to our current environment, then I have a function called copy and vir actually. And then I pass the name of the object. I can pass the list, but I'll be copying all of them. So then I press that, you see all, we copy all those files from the new environment to our current. So then here, up here, you can also see all those files.
Daniela?
Yeah.
There's a couple more questions have popped up.
Great.
Let me show on stage the first one.
Okay, let's see if I can read here.
Can you see that? He wants to know where are the R files used in this pipeline stored?
Okay. Can I answer in a few seconds that?
I'm sure that's fine.
Okay, because I will demonstrate here.
I'll show the other question, and then I'll let you. Yeah. So this person wants to know. Oh, this was the answer. This is the answer from Zhang.
Yes. Okay.
Okay.
Great. I'll show how to handle the R code in that in just a few seconds. Okay, so that was it from the project, the SPR project function. And then to answer that question and show a little bit more details about how to add in an interactive mode more steps, I will go down, because here is like some information about that. So if you want to come here later to read all that, so jump to the 453, the line, and then I will start this project from scratch. So I'll do an overwrite. And then now if you want to check Sol, it's again empty instance of sysaxs list. And then the length, as I said, is 0. Okay, so in order to us to append the first step, I'll go over a few details here. The first one is we have a function, a constructor functions to build this step called sysaxs list. And what this function is expecting is a few details like the step name. So here I can give any name, like I'm giving the trimming. I'm using the same example we just run. So it's Trimmomatic and the index and then the mapping. And then I'm using the targets files, that initial targets I was mentioned in the presentation. So I can open up here. You see this tabular file. It's basically the two first columns are the path of the raw data, and then here I have the sample name as ID for each one of unique for each one of the samples, and then the biological replics here. So it's a simple tabular file for the initial data, the initial targets file. So then what I'm passing here in the targets arguments is just that the path of that file. Then I have all the CWL. Here is Trimmomatic, but it's the same way I just demonstrate to create that on fly. And here is where the magic happens. So the input var is where happens the connection between the CWL definition and systemPipeR. So what we are taking here is a name vector, and then the names of each one of those elements are actually the column names here. So I have file name 1, file name 2, sample name. So that is the information I'm using to build this step. And then the actually element is the variable we define on the input from the CWL. So and then we make this connection. So and before I run this, the other thing I would like to mention is the append step method. So that is taking this object, the cell, the empty container, and is appending this this step we are creating. So let's jump that, and then now we can see, oops, down here, we have the instance of sysaxis list with one step. It's trimomatic. It's pending because I refresh everything, so it's pending the status. And then we have our first step from the command line. So now let's add a little more steps. So the next step, I am on line 485, if anyone is following me. It's to build the index, right? The same thing, that will be a command line step. So I have the append step, the sysaxis list, and the only two things I would like to highlight here. Number one is now targets for the index. I don't have many samples. It's just one, the reference genome. So then, as I said on the presentation, the targets can be optional. And then in this case, we are passing null. And the other thing I would like to highlight, and then here I'm defining the dependency. So I'm saying I'm depending of, in this case, in this example, the previous step, which is trimming. So you pass the name of that. So I'll run that. And now I'll check Cassol again. So now we have two steps. Again, still two command lines. I add one more command line step, and then I'll go to the R step. I forgot to say here, we have one method called cmd list. So here. So then here you can see I actually print all, but you can select just one sample or one step. So in this case, you can actually see this command. We didn't run this command yet. It's just rendered here in the object. So this is also another powerful thing, because you can actually examine everything in your workflow before you run, and then you could have avoid any debugging or any error in front here. And then here is for the index. Then you have one step, and then you see here the command line. Okay, so now I'll jump down to the line 547. And here I would like to add the mapping, right? However, the mapping, I don't want to use the raw data. I want to use the AL files from the trimming, right? So the AL files we create in the trimming. And then here, again, append the step, sysaxis list, and then the step name. And then on the targets, in this case, we are accepting the name of the previous step you want to connect. So here we are building this connectivity between the steps, not just by the dependency, but by the inputs and outputs. So I pass here the name of that particular step, and then we need to inform which one of the out files we want to use. And for that, we can examine the out files. And here, let me print that. So you then you can see here we have the trimming, and then we have this first database where we are expecting 4 files for each one of the samples. So first one is the trimomatic one paired, and the ones we want to use is the trimomatic one pair in the third column, also the trimomatic two paired. So when I am specifying the input variables, now because I'm using the targets from this connectivity from the AL files from the first step, now our name vector will be with the name of that particular column, that file you want to use to that column. So now I'm using here the trimomatic one pair, and then again the same thing here, the variable we define in the input CWL file. So again, here is trimomatic one paired, trimomatic two pair, and all the variables we have defined in this input YAML. So here is actually the magic is happening again, because we are making this connectivity between not just, in this case, is double, because now between the targets file and the input file from CWL, but also with another step, a previous step, which is the trimming. And then down here, I'm establishing the connection. You can have multiple dependencies. So in this case, I'm depending on trimming and also on the high set index. So let's run that. And then now I can examine again. And then we have 3 steps. Again, same thing, all the sub-steps we have defined here, and everything I was describing before. Okay, and then one of the things we can quick look at is if the target's from, let's print this different. If the target's now, it's okay, right? If that connection happens in the right way. So the file name 1 and file name 2 are here because of the first step. We use that file from the disk. However, here on this the mapping, you can see now we are expecting that out file from the first step. Also, you can always check the out file. So here, and then we see all those 4 files and everything. So now I will answer the question. Oh, and then also you can check all the command lines. So here is actually 4 because we have the sub-step. So we have high set and all the same tools, command lines as well. So now I would like to answer that question, is how you append an R code, R base code as any step. So in that case, here actually, to continue our story here, we have a method that will I'll run here, so then it's easy to explain. So the get column, actually I'm getting the column from the step called the mapping, and then I am grabbing the column from out files, and then this specific one, which is the BAM files, because we want to calculate some statistics about the mapping, right? So, and this information now we can pass to a function to calculate that, and then we can also write a table or anything. So notice here between 604 and 7, I have 4 lines of classic R code, right? The only thing I'm separate here by a new line, and that's it. So in this case, instead to use the syslist function, the constructor function, use the line wise. And then you have each line as a new line, each line of the code as a new line, and then you add that between braces, and then the same thing, the step name, and then the dependency as well. So I'll run that, and then we have another step down here. How you can look and make sure everything is okay. We had a function called code line, and then I'm gonna print the whole object. This function will tell you the first 3 steps doesn't have any R code, so it's not a line-wise object. And then the 4th one, it is the mapping. So here you can see the exactly code. So it's very easy to look and then see if there's anything is wrong. Another detail, we haven't run this yet. This is just an expression, and we'll run soon. So now we have all the steps. Also, you can see the dependency. So you can see we don't have any dependency in the first. All the details. Also, another method here to examine this object is to look the steps. So this will give you more information. So steps workflow, then will show like more details about each one. So this is an instance of syslist, which means is the command line. More information here. In the case of this step, you have all the 4 sub-step, and this is the line wise. Okay, so let's run this again. So I'll click here I'm using the run workflow function, and then let's see. this one was really fast because we already have all the results here. So the function was just checking if the expected out file was there, and then we just switched to success. I didn't run anything because I already had that. However, run workflow function has the possibility here, I'm showing here, to force. So if I say I can run this again. In this case, you notice that will take a little bit longer because actually it's running again. So we have the possibility. And then also we can ignore all the warnings, and then also ignore if you have any errors, we are continue. Or you can choose to stop if you have any errors there. Also, you can choose if you want to run just a few steps. If you follow the dependency graph, that will be okay. Everything is here being created. I guess let me go down here. I already mentioned about the view in Vir to look all our objects we create and how we copy them to our current environment. And I also showed before how to use the plot workflow function and how to generate the technical reports to look all the logs. So you can also double check here. And then, as I mentioned in the presentation, because we don't have a scheduler available here, I'm not demonstrating this, but it's available also to parallel this on a cluster. Okay, the last thing I will talk about here is how to export. So we have here an entire object. This is our list container, the workflow container, which has all those 4 steps. And I did this in interrupt mode here on our current session. So one of the possibilities is to export this to our markdown format. So I have this function called sol to our markdown. And when what this is doing is here on our current environment, we have now created this SPR template, which is basically everything we just create live here will be saved in our markdown format. And then now you can go back to the beginning and use the import function again and generate the workflow really, really quick. And the other possibility is to export this to the bash. So in that case, we have the function called sol to bash, and then run, and then you can see pop up here down here a bash script. Is basically each one of those command lines is create here, and then also here for the person who asked about the R code, we are create an R script here. So you can see, and then we are collapsing all the R code is together. So if you have like several steps, which is just R code, we are putting that together in one R script file, .r file. And then I mentioned in the presentation, we have the possibilities to resume and to restart the project. And that is, we are able to do that again with the SPR project function. So if you use the resume through, like for instance, everything crashes, something happened, and then, or you stopped your work today and you want to come back tomorrow, you are not losing anything because everything is saved in that database I was mentioned before. So the next time you open R, you just use SPR project and you resume, and everything you can start from the point you stopped yesterday. The other possibility is to say, oh, and here in the SPR, we also when we use the run workflow function, we can save this RDS with all those objects. So it's also easy to resume because you can say resume true and then load that environment. So load all those files as well, and the new environment you create or in your global environment as well. And the restart option is a little bit more dramatic because you are deleting all the logs, that give you the possibility to delete all the logs and just to import the workflow. And one more very drastic option is the overwrite. So that will like delete everything you have in your log and then also the database and start a new fresh object. So yeah, I guess that was it. Here I have a link to this page up here. So then you can see here more information about the CWL syntax, the project structure, but also here you have all this list is functions that interact with the Caesar's list object. So you have a lot, not everything I could cover here because of the time, but you have a lot of functions to interact and config and replace values and so on. It's available here. And then with that, I will invite my co-worker to go with you to give an overview about systemPipeShiny. Before we move actually to Le to present the overview about the Shiny, does anyone have any questions? Do we have anything in the QA? No? Okay, so I jump out and then Le will sit here and talk with you guys.
Okay. So yeah, hello everyone. So this is Le. So in the next 10 and 20 minutes, I'll be talking with you about systemPipeShiny. So let's just first come to the slides. Where are the slides?
I may close those. One more. Go to the page. That you can find there.
I don't see the slides.
Okay, open here and then go with slides.
Oh, okay.
You can open again, sorry.
Okay, so just briefly say what systemShiny does. First, it does help you to create a workflow and manage the workflow directly from the Shiny app. So it's a web application. And the second thing it does is to help you to do data visualization. So this is the overview of what Shiny, the SPS is. So in the next talk, I will just refer systemPipeShiny as SPS, systemPipeR as SPR to be easy. So then for SPS, it does those two things. I'll explain in a little bit details. So when you launch the SPS app, this is what you see. We'll have different modules. First is the workflow module. Then we have downstream visualization modules, RNA-Seq. And then we have a ggplot module, which you input any kind of tabular data, and I'll help you to create a ggplot. And then we have this custom types, means we can allow you to integrate your own plotting tabs, your own Shiny part to the framework. And last, we have this canvas module, which is a workbench for you to do quick image editing. And all of these features are listed on the gallery when you start the app. And if you are not sure what are those, you can click the links down the image. Then we'll jump to our website. We have very clear menu of what those features are and guidance for you to use those features in the app. And then this is the workflow module. Today I'll demonstrate this module in details. So for here, the slides, I'll just skip everything. But for in brief, it helps you to, in a guided manner, to help you to create a workflow from scratch, to edit the targets file, to create the workflow steps interactively, and then finally you can directly run the whole workflow inside the Shiny app without using any typing any R code or any like command line code. in your terminal. And this is the screenshot of the targets, how you design targets interactively. And this is how you design workflow steps. And this is how you directly run the workflow. It's like RStudio, not RStudio in the app, and you can directly execute the workflow and any kind of R code you want. I'll explain in the demo later. Then we have the RNA-Seq module. And all we want from you is a raw count table and the targets file, the same target file you use to run the workflow. Then we can help you to normalize data, to help you to plot all kinds of clustering methods and make the plots. And then you can do the DAG analysis as well. And we have all the plotting options: volcano plots, offset plots, MA plots, heat map. And also you can download those tables, DAG tables as well. And then we have this ggplot module. Means in step 1, you just give me a tabular file, any tabular file. In step 2, all the columns in that tabular file will be listed here, the first row, and you just drag it to the second row. Then in a second, it will just give you a ggplot back in here. Then we have this next feature is the canvas feature, which you take or you click those buttons, means to canvas. You have seen those buttons in some previous slides. Means we're taking the snapshot or screenshot of those plots and sending a copy to this workbench. Then in this workbench, you can just drag those images to this editing tool. Then you can just start make some edits and make your scientific plots over here. And when you're done everything, you can just download the plot. And also you can upload your own image to the workbench and do some editing. And then, like I said, we'll allow you to build your own custom visualization tabs to append to the dashboard. So it's very easy. You just use this function and we'll provide you a nice template. All you need to do is just focus on the plotting code. We'll handle the rest for you. So all you need to do is just focus on these 3 lines, the ggplot line, or you can replace with base plot code. And then everything we'll give you is a tab like this. So we handle the progress checking, we'll handle the file upload, we'll handle the connectivity to other modules for you. So all you need is just focus on how to make the plot. And then as a framework, SPS allow you to configure a lot of things. So you can see there's a list of all the options you can have. Then that means you can change almost everything you see on the dashboard. Anything you don't want, like you don't want any modules, you can just disable those modules or override with your own modules, or just load your new custom things. Then we also care about the app security. So if you don't want any person to use your app, I don't know why, but if you don't want your friend to use your app, you can enable the login. So by enable the login, only some the certified users with the accounts can use the app. And then you can add a loading theme before the people are seeing the login screen. And it will become useful if you enable the login, the admin will take in place. So it's a customized hidden URL you can use. Then you can customize the URL as you like. Then when you enter the URL, you will be going to this page, and you need to have an admin account to log in this page. So in this page, you can just see the server status of everything is running in your server. And you can also control the users from here, like add a user, change user, delete user, or other activities you want over here. So in the end, last slides here, is showing SPS as a framework. We have also some supporting packages built inside the SPS umbrella. So we have SPS comps, the jar, and SPS util. So those packages are good to use for your own custom Shiny apps and also R markdowns. So we're not only showing a nice Shiny app to you, also want you to be successful in your own development of Shiny apps and R markdown writing. Cool. So the demo session. You have been disconnected. Did you just log me out?
No.
So it appears we have some brief technical difficulties.
It says I'm logged out. I disconnected.
So if everyone can just hang on while our speakers get reconnected. Do you guys need some help? I can hear you. Uh-oh. Oh, I see. Oh, I see. It looks like Daniela is reconnecting. Are you back?
Yeah, so
good.
So we have some disconnection over here. So I can share
Welcome back.
This window, right? Share a window. Absence window. So can you see my screen okay?
Also, I wanted to point out there's a question that's popped up into the Q&A. Can you read it? Or I can show it on the stage if it helps. Here, how's that?
So I guess.
Yes. Turn off your mic.
Okay.
I guess I can answer that. Yeah, if CWL.
What was that, Daniela?
They can't hear you.
Huh?
They can't hear you. Come here to talk, maybe.
Yeah. Okay, so I'm here close. So yeah, because we are using CWL allows to have Conda and also Docker or Singularity. So that because we are using CWL, we can be able to use those environment in management.
Yeah. That's great. Thank you.
Okay. All right. So next, I will just demonstrate the Shiny app in the RStudio server we have. So first, we just clean everything. We may want to try to change the working directory back to the home directory. Just change it back. And then what we start is just by typing the systemPipeShiny. Oh, I missed an S here. And you type SPS init, means create a new systemPipeShiny project for me. So in 1 second, you will have the project over here. So at this stage, the app is deploy ready and run ready. So that means you can just run the app and deploy it as well. So you click this button, it will automatically deploy for you. And if you run here, click here, it will run. So I always select to run external, means opening a new tab browser. I don't want to save that file. And here are all those app options over here. You can change, like I said, in the list. But here I will just use everything as default to continue. So at the first time, if you use the workflow module, it takes some time, like 10, 20 seconds to load the module and everything. And after a while, you will see the whole app is started. Then for this demo, I'll focus on the workflow module. And since we don't have much time, so you can just play other modules by yourself. And for here, this is the workflow module here. First, what we see is we are have different steps to create the workflow. So in the first step, you need to generate a workflow environment. So you can use example or RNA-Seq or other things. So in this demo, I will use RNA-Seq to demonstrate a whole workflow module, the whole workflow, and I will directly execute example in the second run. That's because to run the whole RNA-Seq will take too much time. So running the example will save a lot of time. But to design the workflow, we can still use the RNA-Seq. Just before execution, we'll change to the example. So by clicking here, we'll create the workflow environment for you. You can see where are the files located. Then you'll have these targets here, this target file. You can choose the default by the template, or you can upload your own targets file. Then you can just edit any the path of all the files here as anything you want. And you can also select some rows and right-click to remove the rows, add rows. Something happened. Let me reconnect back to the session. So we're here. So you can see here is the file, the folder we just created. Since we disconnected, let me just recreate the thing. So we use the RNA-Seq. Here's the target step. We can right-click to insert the change column to remove anything. For here, I just use default. So on the left is the statistics of your targets. So you can see the sample size column, and you can choose whether those files exist by clicking here to check. See, I have all the files for the first column, second column, and third column is just factors. So I shouldn't have those files. See, I have missing 18 samples here. So when you are checking everything and you are satisfied with the targets, you can just go to the next step by clicking here. And when you are seeing RStudio thing, we are importing the steps to the saw object, like Danny just talked about in the first part. So after everything done, then we can go to the next step to design your workflow. That's over here. So on the left is your workflow designer. That just enlarge and minimize the bar on the left. So you can see all your workflow steps are on the left, and your dependency plot is on the right. So on the left, you can just simply drag and drop any steps to anywhere you want. So it decides how the order of those steps will be executed in later when you actually run the step. So if you change order to a wrong position, that means your dependency is messed up, because this step is depending on the heat map step. So you can click the config button, which is a cog-like button. So you will see this step has dependencies messed up. So you need to fix it by choosing the previous step here. So you can see the previous step is get go annotation. So come here to choose Go annotation. And you can choose multiple steps as dependencies, but here I'll just choose one. See, everything is fixed. And then you change the session info steps after the Go annotation steps. This is how you fix the dependency. Then you can always just regret and say, oh, I did something wrong. I just go back one step, go back 2 steps. This is the initial saw we have. Then for configuration for R step and sysarg steps, means command line step, they are different. For example, this is R step. So you can see the R code we're running over here. You can change any R code you want. And for sysarg steps, you are only allowed to change the step name and dependency. All other files requires the CWL files to render the sysarg steps. So we don't allow you to change anything. We just display everything here. So for example, the command line will be running, and the targets table and out file table that will be generated. Then you can create a new step by clicking here. So for R step, it's very easy. You just type your R code here, the step name, and step dependency. By default is depending on the previous step, and append as the last step for you. For example, I go 1+1 here. In a second, you will have a new R step over here. And you can see this is my new created step. And then you can create a sysarg step, a command line step as well. So the basic arguments are similar to R step, but for the CWL arguments, you need to prepare 3 things. So first is the targets file, or you can inherit targets from a previous step, meaning the output from a previous step can become the input for the next step. So if you choose previous step, you will choose the step name you want. And then if you want a new targets file, you can just upload over here. For using this is the first thing I would do is to just select the desired CWL file and the CWL YAML file you want. So normally they are in pair, so you just select the paired file. So I just directly hit the render button. You can see the base command rendered by these 2 files. Then we can start parse, use the targets file to replace those variables inside the base command. And for example, I choose mapping step as the dependency to get the columns as targets. And over here, you can see we need to replace 2 variables. One is called a string, one is called the sample. So here, you can choose which column from this targets table to replace the variable. You can use for string, let's just use factor. For sample, we have a sample name. And if you just re-render everything, you can see everything is being re-rendered and get correctly parsed by sample by sample. So you have all the factors, all the sample name, and parse the command. If you are satisfied with everything, then you can just save here. You can see this step is added. And then we have the step added to the last. Of course, if you don't want any step, just drag to the trash can, right? And this step is deleted. And then after everything, you can save the whole workflow and go to the final running step. So for here, like I said, I will change to the example workflow and run the workflow. That will save us much time. So first, you just refresh the Shiny app to give you a new session. New Shiny session. There's no need to restart, just refresh the tab. So here are just some interactive guide you can play with. They're over here. Now we'll have notifications over here. once in a while we'll send notification to users, like we update the package, we have new features, for example. And then let's just use example, create a new example workflow. And this workflow is very easy. And the targets, just use default targets. I'll explain the code of the example workflow a little bit. So first, we just export the iris dataset into a few CSV files by species. Then we just gzip those files. Then we just unzip those files again. And in the last, we import those files back to R and do some statistics, like binding the data frame and do standard deviation and mean. Then finally, we'll make a ggplot. Then I can show you how easy it is if you just run your custom steps. Like I have a new step, it's 1 + 1. All right, remember this step 5 is added here. I save. I'll go to the last part. So over here, you can choose to run directly in SPS or just download everything you created and run in a different place. So this is the running space. You can see we have the initial workflow. There's nothing has been run, so it's all black, the steps, and you don't have any log displayed. We haven't run anything. And on the left top is the source code you want to run. And I can, by default, I'll configure the code for you, but you can run any R code you want. And the bottom left corner is the output. I'll just run the steps. And on the right part is the plot you make. Will automatically capture any plots you make in this session to this place, and you can see those plots. So it's you first need to load systemPipeR, so it takes some time. But after loading, it will just take 1 second because this step is very easy. You can see everything is running successfully, and you can see the plot becomes green. We have all the things successfully run, and all the file samples pass over here. And you can switch to the log to see all the things, the code, the standard out, and other things in the log file. So I can also run any arbitrary R code I want. For example, I make a plot, right? Over here. You can see it's automatically captured over here. And this is pretty much about the workflow module. Then this module connect with another great visualization module in SPS, which is the Canvas module. So by connecting to that module, you need to send screenshots to that module. So first, you just click this button, for example. I'll just send 2 screenshots. Then if you close here, and then after you run everything, you can just download all the things as a bundle, like I said. So if you click the first option, it means you will also translate the module, the workflow you have run to bash script. Also can translate to a R Markdown template file, so you can share this file with your friends. Okay, so it is downloaded. Okay, but I guess you cannot see the downloading because I'm not sharing that tab. But you can try it by yourself. So I just directly go to this module, the Canvas module. So remember, I hit the button twice and send 2 screenshots. Then those 2 screenshots will become available in this module. Then what you only need to do is just drag those plots you want to the workbench and resize, do some resizing, change the angle, and copy paste. So like Ctrl-Z, Ctrl-V, those short keyboard shortcuts are working. And you can also add some text. My figure 1, for example, and change color and do some other things as well. And then in the end, when you are satisfied, you can just download the whole image to your local. So this is like some core functionalities of systemPipeShiny. But just remember, we have more things to play with. We just don't have the time today. So please try by yourself those cool features. And here are the resources we have. And here's our team. And if you have any questions, just let us know. And please just check our website. And we have all the menus. I can show you quickly. Here's our website. We have all the detailed menu for systemPipeR and systemPipeShiny. And I think, yeah, that's pretty much about today's presentation. And we really thank you for attending the workshop. So if you have any other questions, just let us know.
Thanks, guys. I think we have to close now because their session is going to start, and I actually have to go over there, and I actually can't leave until I close this one. Thank you.
All right. Thank you. Bye-bye.